Let's continue here with the genetic code. So we said earlier that we have a 4-letter alphabet, A, G, C, U. And those 4 letters, they need to be put together as 3-letter code words. So we have then 64 possibilities. So it would be 4 times 4 times 4 possibilities to make 3-letter words with a 4-letter alphabet. And those combinations are seen right here. So if you are taking a look at the genetic code, you will see all 64 possible code words spelled out right here. You have first, second, third position, putting these code words together, and then you will see that you have 64 possibilities to make these 3-letter code words. Now, 3 out of those 64 code words, they are stop codons. That means they are not associated with an amino acid. So that would be this one right here, UAA, UAG, and UGA. These are all considered stop codons because there's no amino acid that's associated with them. Then there's another one here, the AUG, that is the start codon, and it specifies a methionine. It's the only 3-letter word that specifies methionine. It's considered a start codon. So out of the 64 3-letter words, 61 specify an amino acid, and then 3 are stop codons, and that's what we had in the previous slide. There you have these 61 sense codons that specify amino acids. Now, there are only 20 amino acids, so it's not a 1-to-1 ratio here, and that leads us to the term that the genetic code is degenerate. I don't really like to use that word too much. It's so negative, but another word that you can use for that is that it's redundant. So there is a redundancy in the genetic code. That means there are several codons that specify the same amino acid. So you can see that here, for example, let's use the first one right here. UU and UUC both specify phenylalanine. Or leucine here has even 6 possibilities. UUA, UUG, CUU, CUC, CUA, and CUG. They all specify the same amino acid, leucine. Okay, so that's known as the redundancy or the degenerate nature of the genetic code. The genetic code is universal across all species of life on planet Earth, and there are very, very, very few exceptions. There are some variants, but for the most part, the genetic code is considered universal across all forms of life on planet Earth. So now we're ready to move on to translation. And translation of the messenger RNA will begin with the start codon AUG. So that's the one that we just had here. This is your methionine. AUG is your start codon. And once we are reading the first codon, this AUG, at the ribosome, then we will be adding amino acids code word by code word, reading the codons of the messenger RNA and translating them into amino acids specified in the genetic code until we reach one of the stop codons, which is UAA, UAG, or UGA, and then the translation will stop, and then the protein just needs to be processed further. But as far as translation is concerned, we have now translated from the nucleotide-based code of the messenger RNA to the amino acid-based code of the proteins. Okay, so let's take a closer look at how translation works. We need a mediator molecule that's called a tRNA molecule that carries an anticodon that matches the codon, the RNA, messenger RNA codon. So we need an anticodon that matches it by base pairing rules. And then on the other end of that tRNA molecule, you're carrying an amino acid. And then you just need to connect the amino acids one by one with peptide bonds. So here's a diagram of how this works. So at the ribosome, we have 2 subunits, the small and the large ribosomal subunit. They come together, it's almost like a sandwich. And then you're threading the messenger RNA molecule sort of between the two subunits of the ribosome, and it starts reading it. And so it will scan the ribosome will scan the messenger RNA until it finds the AUG. At the AUG, the start codon, the first amino acid will be delivered, and so the anticodon of this particular tRNA molecule, so the tRNA is this clover leaf shaped molecule. It has the anticodon on one end, and then here the amino acid on the other end. So the anticodon has to match the code word. So matching to AUG here on this start codon, of course, is UAC. So this tRNA molecule carries a UAC anticodon that matches here with the AUG on the messenger RNA, and it delivers us a methionine. So here is your first amino acid. Once this has happened, then the ribosome will scan the next triplet word, the next codon. And here we have a UUA as a second codon. Now the ribosome will wait until an amino acid, a tRNA molecule with the correct anticodon will come by. So for UUA, the matching anticodon would be AAU. So U with A with U. So once we have an tRNA molecule that matches the codon here with the appropriate anticodon, then the ribosome will then get this second tRNA molecule in close proximity so they can deliver its leucine, and then we need to form a peptide bond between methionine and leucine. Meanwhile, once that has happened, then we can discharge that tRNA molecule that has delivered its methionine. So let's move on. So now here's the next step. So here's our leucine. Now we just need to form the peptide bond between these two amino acids, and now we can get rid of that. We can discharge this first tRNA molecule. It's going to move into the E site, E for exit site, and so it's no longer carries a methionine right here, and it just will fall off. Then we need to move the leucine carrying tRNA over toward the methionine carrying a start codon, start tRNA molecule was first, and so then we have room for another tRNA molecule that has to match the code word. Next code word is GGU. The matching anticodon is CCA, and now this amino acid will deliver, this tRNA will deliver the amino acid glycine, and now we just need to form a peptide bond. Then we can move everything one place over, and now this tRNA molecule would then leave, and we can add the next one. So go step by step until we hit the stop codon. So here, the next step here, so we need to form the peptide bond between leucine and glycine, so that's going to happen. This one here, this was the tRNA that carried the methionine, so that one is leaving now, and now then we need to move everything over by one place, which has happened here. So now we're discharging the next tRNA molecule that has delivered its amino acid. So here we're one step further. So here the glycine was bonding with phenylalanine. Now we're here, and we are discharging the one that carried the glycine amino acid. So I think you get the idea. We're going to continue this until we hit the stop codon. So once we hit a stop codon, there's no tRNA with a matching anticodon that would carry an amino acid. So we now are reaching the end here of our translation, and the ribosome will then release sort of this polypeptide chain and discharge all of the tRNAs that have delivered their amino acids. Here is the last one is leaving, and now we have the new protein that just needs to be folded and sometimes cut in specific places, but the primary amino acid sequence is there now, starting with the methionine, and then here we're ending with an arginine. Okay, so In bacteria, one cool thing is translation can begin before transcription is complete. Since we don't have to deal with the nucleus, we don't have to move anything. So really, as you're making your messenger RNA, you can already start translating into protein. So everything's faster and very efficient in prokaryotes, because you don't have to move messenger RNA from the nucleus into the cytoplasm where the ribosomes are. So conveniently, in prokaryotic cells, everything is right there. And a lot of times you have literally an assembly line of ribosomes that are making multiple copies of the same protein. So once you have the messenger RNA, then you can attach multiple ribosomes. Those are these little red dots right there. They're all working on it. So here's your DNA. You're making a transcript, and that would be here, right here. So you have the transcript that's forming, and then you're attaching multiple. So in blue, that's the transcript, the messenger RNA right here and right here. So you're making these transcripts, and then you're attaching multiple ribosomes. You can see that you can make a bunch of the protein product in very little time if you are using an assembly line method like this. So here, one more time, the overall process we're talking about right now is what we call the central dogma of molecular biology. And that will be, we're going from DNA to messenger RNA via transcription, and then messenger RNA to protein via translation. And since we don't have to deal with a nucleus, everything is very efficient and fast and conveniently easy in prokaryotes. Now let's take a look at transcription in eukaryotic cells. And there you're dealing with whole new dimensions of complexity. For one, you have that nucleus that you're going to have to deal with, and then the messenger RNA product would have to be moved from the nucleus into the cytoplasm for translation. But even just starting out with the transcription itself, you're going to have a lot more complexity to this. Okay, so for one is that in eukaryotic genomes, there's a lot more non-coding sequence present. In E. coli or in prokaryotes in general, you have hardly no intron sequences. So that means everything is pretty much either regulatory region. It's a little bit of repeat, but mostly you have just DNA that is either coding sequence or regulatory sequence. Now in eukaryotes, we're adding something called introns. Introns are large amounts of DNA that don't code for protein. And what makes it worse is that it's not just sort of patches of intron and exon sequences that are separate, but the introns are part of the genes. That means they need to be cut out. If you want to put your genes together to a readable messenger RNA that can be translated, you need to get rid of all of the intron sequences, and that is not so easy. So there is a process known as splicing. And basically, what you need to do is, let me go to the next picture here that will explain it. So here would be a eukaryotic DNA has altering exon and intron sequences. And by the way, it may not look like this here, but intron sequences, there's about 10 times more intron sequence in eukaryotic genes as opposed to exon sequences. It seems almost like as if there's more exon sequence, but that's not really true. So, but just in principle here, what you can see is that you need to put the exons together. So you need to get rid of these intron sequences. You know, cut that out. This one and this one, they have to go. Now, you can't make a mistake, because if you were to cut at the wrong place, even if it's just one nucleotide the wrong place, then you're going to end up with a frameshift mutation, and that would be terrible. So you need to be very careful when you are removing these sections. So here we have the DNA, and you have to make an initial transcript. So here's your RNA transcript, and it contains the introns still, because that was part of what the RNA polymerase was just making a copy, an RNA copy of this stretch of DNA. So now the introns here again, so they're still part of the now RNA, they need to be removed. That's accomplished with these snRNPs that we had in the previous slide that I kind of skipped over. So these small nuclear ribonucleoproteins, snRNPs, they will help with the splicing process, which is a very complicated kind of process. But anyway, so once we have these introns removed, then we need to put the exons together, and we can do this even in alternative ways. It gives us some options, but then the messenger RNA needs to leave the nucleus via these nuclear pores and end up in the cytoplasm for translation.